I want to thank Rod, my very good friend and special friend from Charlottesville, UVA, from the long old days, and all organizers of this course, particularly Linda and Megan, for having us again in this yearly neuroanatomy course. And here in my side, I want to thank my secretary, Camila, who has to help me even to turn the computer on and off. The course will have this format of 3 people speaking. Some of the lectures will be in 3D. We'll tell you when to put your glasses. And we are very fortunate to this year to have here Professor Hugo Ture. We'll talk to him later. Well, unfortunately, we're not able to be all together this time in Seattle, but fortunately, we can do it through Zoom. And I'm very proud to announce that, as far as I know, this is the first course that we're going to do stereoscopically using 3D glasses and showing some 3D images, as you're going to see shortly. I want also initially to thank Richard Paraga, who was our fellow in Brazil at Professor Evandro Oliveira Lab a few years ago, who did most of the sections I'm going to show. One also to thank my son Eduardo, not only for his dissections, but mainly for his support with the 3D. Well, we're going to be talking about the brain architecture, particularly of the telencephalic structures of the cerebral hemispheres. But let me start emphasizing that neuroanatomy can be studied and has to be studied through very different dimensions. But morphology is mandatory, is crucial for the understanding of any other dimension of neuroanatomy. So when you see this lesion, for example, you initially recognize the surface of the brain, and then you recognize the anterior horn of the ventricle by its shape. And you have to know that the lateral wall of the anterior horn is the head of the caudate, which harbors this lesion. So this is an information you have to know. And then when you go to surgery, you have to do it as much as you can through the natural spaces of the brain. And when you get in the ventricular cavity, for example, if you see choroid plexus, you have to know that you are in the body of the ventricle, not in the anterior horn. So you tilt your microscope, see the anterior horn, and again, you have to know that the floor is the rostrum of the callosum and the pillar of the fornix, and the lateral wall is the head of the caudate where you have your lesion, and you can then remove it. So, as you see, we are dealing mostly with spaces and shapes in order to recognize structures, both in our image studies and in our surgery as well. That's why I like to call this lecture Brain Architecture. Now, I'm going to start showing some regular 2D slides about some embryological and evolutionary and embryological tips that will help us very, very much in order to understand the brain architecture, namely the shapes and the space that these structures occupy. Well, as you know, creation of men was not so miraculous as here beautifully painted by William Blake. According to our current knowledge, it took 1 billion years for us to have the first unicellular element, for the first unicellular element to appear. And then it took almost 3 billion years for the first vertebrate to arise, which were these primitive fishes 400 million years ago. During all this last 400 million, the CNS underwent an explosive evolution, an explosive development, until the emergence of man, of our humankind, only 160,000 years ago. Throughout all this evolution, more complex neural structures were progressive developed. And in primates particularly, other than the increase of the brain as a whole itself, ultimately there was a bigger increase of the prefrontal areas, as you can see here. The brain did not only increase regarding its relation to the body, but also in relation to the prefrontal area that increased more than other areas. Overall, in this last 3 to 4 million years, the brain did increase 3 to 4-fold. And this increase, interesting, was more done due to connections than of neurons, and more increase of short connections of new fibers of the cerebral hemispheres. Now, parallel to this, development of complex structures, it's very interesting to see that some structures did an evolution. They turn into vestigial structures. And a very good example of this is the anterior commissure. That was the main commissure for ancient reptiles. You see that nowadays, just 5% of these fibers in monkeys and 1-2% in humans. And to see how important this anterior commissure probably was, you see that the anterior commissure still now, it has 3 times more fibers than the optic nerve, that it's our main cranial nerve. And its density is more than twice the density of the corpus callosum fiber. So it was a very, very important commissure for old reptiles, and now it's just a vestigial structures. So these structures are there, and they make neuroanatomy to be more complicated to study. Now, in order to understand the development of the main structures of the CNS, let's take a look at this very simple but very didactic model called the Triune brain theory that was proposed by Paul MacLean, neuroanatomist of the MIT in 1970. The most primitive vertebrates, these primitive fishes we were talking about, that was 400 million years ago, more or less, they had a very linear and simple CNS. They had this very linear segment, which we'll call the spinal cord, and then they have a very simple brainstem that we will call as having this structure that is, we'll call a hypothalamus already, that was responsible for the control of its vegetative functions, of its autonomic functions. And then it had this very old structure that we'll call striatum, that was responsible for the control of their very, very primitive movements at this time. Now, about 300 million years ago, when they turned into amphibians and then reptiles in order to reach Earth, they had to develop an structure that allowed them to perceive this new world. And they did develop the rhinencephalon, they did develop the olfactory system. Now, they needed a structure that could store these olfactory memories that they were achieving, okay? So they developed a trilaminar structure that we're gonna be calling the hippocampus. And then they had to develop another structure that could drive their behavior according to what they were smelling, to drive their behavior, which was the amygdala. So these 3 structures did arise together, and they were disposed around the stop of this brainstem, and that's why they are called limbic structures, because limbic in Latin means around, means ring. So that's why they came together. They are studied in the same chapter of neuroanatomy, and they are called limbic structures. The olfactory system structures, the hippocampus, and the amygdala. Now, when the mammals came around 100 to 200 million years ago, a new structure had to be developed in order to receive all the new sensation and sensorial inputs. So this structure was the thalamus. And together with the thalamus, the neocortex was developed in order to distinguish this new sensation and sensorial inputs. And together with the neocortex, the corpus callosum was developed. So these structures did arise together with the mammals. So you can see that this was a progressive development throughout this evolution that took all this million years. Now, it's very interesting to see that at least at its major steps, embryology sort of repeats the main steps of phylogeny, of all this evolution. And Also interesting to emphasize that the bending and folding mechanisms towards and around the center were the main mechanism that nature found in order to save space. And these structures were then built around the center of the brain, which is given by the thalamus. So the thalamus is the morphological and the functional center of the brain. Now, you remember that the neural tube had initially 3 vesicles, and these 3 vesicles turned into 5 vesicles, since the prosencephalic vesicle, which is the cranial one, divides in 2, and the upper one will later develop in 2 other vesicles. So the wall of this neural tube will originate all the neural structures, while the inner compartments of the neural tube will develop the ventricular cavities. Now, since the upper part of the prosencephalic vesicle divides in 2, each one of these sides will become the lateral ventricle, will bend around the center and assume this C-shaped format, while the lower part of the prosencephalic vesicle will turn into the third ventricle. So the wall related with the upper part will originate the telencephalic structures, while the wall related with the lower part will originate the diencephalic structures. Now, it's very interesting that the upper part of the neural tube will turn into the lamina terminalis, which is the anterior wall of the third ventricle. So the third ventricle is related all with the encephalic structures, but its anterior part, which is the lamina terminalis, it's indeed a telencephalic structures due to this movements of bending and dividing and so on. Now, finally, the lateral ventricle will end up as a C-shaped format, as I already mentioned, wrapping the thalamus, while the third ventricle will be between both thalami. So if you understand this evolution, you can understand why these structures have this shape. Another very important, interesting thing to have in mind and to know is that the hippocampus, as already said, initially was at the top of this very primitive, very old brainstem, and then it did slip around the center, which is the thalamus, and it left a tail when it was slipping. So this tail that was left became the fornix. Now, since this took place at the same time that the cerebral hemispheres and the corpus callosum were being developed in this very early mammals, the vestigial hippocampus remained along the upper part of the corpus callosum as the indusium griseum, which is connected to the hippocampus through the gyri fasciolari, the so-called gyri fasciolari. So if you understand that the hippocampus did slide here and leaving the tail, which is the fornix, at the same time the corpus callosum would be developed, we are going to understand why these structures have this very, very peculiar anatomy, this very peculiar shape. That's why I tell you that understanding of this evolutionary steps help us very much to understand the shape of these structures. So finally, the hippocampus together with the fornix, which is called the hippocampal formation, will be just docked around the inside of the ventricular system, again with the C-shaped format, having the amygdala just anterior to the head of the hippocampus, as we're going to be seeing later with more details. Everything, of course, around the thalamus. Now, another very interesting and very important tip is to understand that when the mammals came again and neocortex developed together with the corpus callosum, of course, the projection fibers have to be developed also. And the development of these projection fibers did split the corpus callosum, it split the old single striatum, okay? Now, and all these structures also did bend around the center, which again is the thalamus, assuming the C-shaped format. Now, since the corona radiata continues with the internal capsule, and the internal capsule have this fan-shaped format, it turns out that only the dorsal striatum was divided. And the dorsal striatum then originated medially the caudate nucleus, and lateral the putamen, of course. But given this fan-shaped format, the ventral striatum, the anterior and basal part of the striatum, was not separated. And so the ventral striatum became as a single region and harboring the so-called accumbens nucleus, the nucleus accumbens, and we'll return to this nucleus later on. And of course, everything again with the C-shaped format around the thalamus. You see that this bending and folding process around and towards the thalamus now was responsible for this issue that most of these deep structures, the inner structures of the telencephalon, have the C-shaped format. Now, let's now put our glasses, okay, and go to our part of the lecture in 3D. I hope you are all using your glasses, and we can see here very well the brain already dissected, and we know that the thalamus is exactly in the center of the brain, as we already talked about. We know that the lateral ventricle has the C-shaped format, and everything that it's anterior to the thalamus is the anterior horn or frontal horn. Everything that is superior or upper to the thalamus is the body of the ventricle. Everything that is posterior to the thalamus is the so-called atrium of the ventricle. And inferiorly to the thalamus, we have the inferior horn or the temporal horn. The posterior horn or occipital horn is just a posterior extension and very variable extension of the atrium. Some people don't even have a very big, a very significant posterior horn. So the atrium divides the ventricular cavity. So let's build the brain from the thalamus and understand all these structures. The thalamus is pretty much a well-defined structure, an oval shape, and it's continuous with the optic tract, as you know. And the main thing that we're gonna be talking here, the first thing is that we have to understand the thalamus as the head of the brainstem. Morphologically, both thalami belong to the brainstem morphologically. They are just like the brainstem having two heads without any fissure in between. The thalamus is completely continuous with the midbrain, as you can see here. I forgot to mention that while the thalamus is a very well delineated structure, the hypothalamus is more a region. It's just like a U-shaped region that comes with the upper side, one side, lateral side, and then the bottom, which is, as we're going to be seeing later, the floor of the third ventricle. So while the thalamus is very well delineated, is a structure, I understand the hypothalamus as being mostly as a region, which takes the both sides of both brain hemispheres. Now, the geniculate body of the thalamus, of course, are prominence of the thalamus. You can see here the midbrain, the lateral mesencephalic fissure, and the medial geniculate body is very, very round. It's very easy to be seen in this in the brain when you dissect the brain, and it's always at the top of the lateral mesencephalic sulcus. The lateral geniculate body is more lateral and more anterior, and it's more flat. Sometimes you don't recognize it very well, and of course it is continuous with the optic tract, that it's continuous with the chiasm. But again, let me emphasize here that the thalamus is again the head of the brainstem. There's morphologically, there's no fissure in between. It's pretty much continuous. So when you come back to this slide now, you know that the thalamus is the head of the brainstem. It's in the center of the brain, so the brainstem will be coming pretty much from the center of the brain underneath the thalamus. The first structure we're going to be putting around the thalamus here in the center that we already mentioned is the hippocampus and the fornix. The hippocampus together with the fornix and the dentine nucleus we're going to be talking about shortly, all together some authors call as the hippocampal formation. The hippocampus has a head, has a body, and has a posterior part, a tail of the hippocampus. And the hippocampus is so old that the white matter is in the external, it's outside, just like in the brainstem, just like in the spinal cord. It's cortex, As we already mentioned, it has 3 layers. And the hippocampus is part of the parahippocampal gyri. The parahippocampal gyrus is called parahippocampal gyrus because it was developed along evolution along the hippocampus. Well, this outer surface of the hippocampus, which are the axons of the inner parts of the layers of the hippocampus, are the axons that get together and built the fimbria of the fornix initially. So this very thin initial part of the fornix is called the fimbria. Then posteriorly, we have a more flat part of the fornix, which is called the crura, and then it becomes very cylindric. It becomes the body of the fornix, and then the pillar or column of the fornix that will end in the mammillary body. The head of hippocampus is also called pes hippocampi due to this shape of that looks like the head of a lion, as some authors mention, and has anteriorly the amygdala. As you can see here, the amygdala is not only anterior to the head of hippocampus, but cover a little bit the head of hippocampus. Here is medial, here is lateral, here is anterior. So we can see that this lateral part, this medial part of the parahippocampal gyrus, it's very flat and it's called subiculum because subiculum means the bed. It's both the bed of the hippocampus itself and it's also the bed of the thalamus that will be resting here. And of course, you can see here the fimbria coming here. You can see the dentine nuclei that are very related with the hippocampus. Indeed, we call this prominence the right name for this prominence is Amon horn, and hippocampus is the name that we have to give for both the Amon horn and the dentine nuclei together. And here again, you see the fimbria, and of course, this flat surface is the subiculum. Here, seen from anteriorly, you have here the hippocampus cut. You can see that it has 3 layers, and the outer layer are the axons of these neurons that are in this these inside layers here. And you can see again that these axons, they build the fimbria of the fornix. Medially, we have again the dentine nucleus, and then we have the subiculum. This is all parahippocampal gyrus, and parahippocampal gyrus has laterally the fusiform gyrus, and the deep sulcus that separates the parahippocampal gyrus from the fusiform gyrus, of course, is the collateral sulcus. It's a complete sulcus. It's a very deep sulcus, almost reaching the ventricular cavity, as you can see here. Okay, so this is the hippocampal sulcus that separates the whole hippocampus again, which is the Amon horn and the dentine nucleus, from the rest from the subiculum. Okay, while the hippocampus is archicortex, the subiculum is already paleocortex. It's a newer cortex. And here you can see here already the floor of the inferior horn and of the atrium. We'll be talking about this a little bit later. If we lift the fimbria, we can see better the dentine nucleus. And this is the fimbria dentate sulcus, where sometimes you have the PCA, the posterior cerebral artery, coming until inside this fissure here. And again, you have here the subiculum. Now, both fornix are united along the midline. They get together. So both bodies of the fornix are together here. And of course, they go to each side of the mammillary bodies. And both of them, of course, have the amygdala anteriorly and covering a little bit the most anterior aspect of the hippocampus. Now, the hippocampus then is inside this parahippocampal gyrus. We're going to be talking more about this gyra in the forthcoming lectures. But the parahippocampal gyrus is a very interesting gyrus that looks like the stem of a tree, that it's mainly constituted by as a continuation of the lingual gyrus, as we're going to be seeing later, and of the isthmus of the cingulate that comes right here. And here you can see, of course, the calcarean fissure, which is very, very deep. Anatomy is definitions. Anatomy is nomenclature. So we consider the parahippocampal gyrus everything that comes underneath the beginning of the calcarean fissure here. So from here superior, you have the isthmus, and from here inferior, you have the parahippocampal gyrus. And of course, you have here the uncus, and you have here the uncus sulcus. The parahippocampal gyrus also receive some contributions of the splenium, as you can see here. And again, you can see the fimbria, everything going around the thalamus. When you look at the parahippocampal gyrus from the base, from the bottom, we have to see, remember that the uncus has this triangular shape here with this anterior aspect facing the carotid cistern and its posterior aspect facing already midbrain and with apex 9 now. And regarding this architecture, what I want to emphasize now is that if we divide this triangle in two, the anterior part of the triangle harbors the amygdala, while the posterior part of the triangle harbors the head of the hippocampus already. So if we are going towards the amygdala, for example, for the neurosurgeons, a transcranial approach would be good enough. But if we want to go to the head of the hippocampus, we'll have to get inside the ventricle somehow. So again, this is a triangular shape. If we divide this triangle in two parts, the anterior part harbors the amygdala and the posterior part harbors the head of hippocampus. I already emphasized that the amygdala is anterior to the head of hippocampus and covers a little bit the head of hippocampus. And if we cut the anterior commissure, we're going to come back to this dissection later. We can see, just talking about the amygdala right now, we can see that the amygdala stands superiorly and blends with the globus pallidus. We come back to this beautiful dissection here, the anterior commissure that runs inside the so-called channel of Gratiolet. But at this point, let me just emphasize that the amygdala extends superiorly, blending with the globus pallidus. So in this dissection here, we can see here the base, this very dark part of the brain here, which would be the base of the putamen, and just more medially, the globus pallidus. So the amygdala will come right here and will continue superiorly with the globus pallidus. Again, we have here the hippocampus, and we have here the amygdala, just anterior to the hippocampus, and with this amygdala going up towards the basal forebrain, the most lateral aspect of the basal forebrain, and then we'll have a fissure in between the fornix and the thalamus. And since we have this fissure is the choroidal fissure. So the choroidal fissure will start just posteriorly to where this part of the amygdala will stand up superiorly towards the lateral aspect of the basal forebrain. And this most anterior point of the choroidal fissure we call the inferior choroidal point. Here we have the anterior choroidal artery coming in, and we have the inferior ventricular vein going out. The fornix go around here, the thalamus. You can see here the very flat cruda of the fornix, and then the cylindric aspect of the body of the fornix, and then you have the pillar of the fornix that gets inside the brain. When it gets inside the brain itself, underneath the thalamus, we'll see later, inside the hypothalamus, of course it closes the choroidal fissure. Now, before getting in here, the body of the fornix sort of detaches from the thalamus and enlarges the choroidal fissure, and then gets inside the brain and closes the choroidal fissure. And this enlargement here of the choroidal fissure is the interventricular foramen on Monroe that will go to the third ventricle that would be between both thalami. So the choroidal fissure comes from the inferior choroidal point to the foramen on Monroe. This is a lateral view, of course. Let's see now a medial view. If you see medially, you can see again the crude of the fornix going around the thalamus. This is all the thalamus here. I forgot to emphasize that the posterior part of the thalamus is called the pulvinar of the thalamus. And you have here the cruda, you have here the body, and then you have the pillar of the fornix getting inside the brain itself, going towards the mammillary body. Of course, this enlargement of the fissure here before the fornix getting inside the brain is then the foramen on Monroe. Of course, this is the midbrain and this is the aqueduct. we can usually see very well a deep sulcus that comes from the foramen Monroe until the upper part, the opening of the aqueduct. And this sulcus is called the hypothalamic sulcus because it runs just underneath the thalamus in the inferior and anterior aspect of the thalamus. So superior to the sulcus, this is thalamus. Inferior to the sulcus, this is hypothalamus. That's what I say, that hypothalamus is a region. Okay, now, so the posterior wall of the foramen Monroe is thalamus. And you have here 2 or 3 prominences, which are the anterior thalamic tubercle, which harbors the anterior thalamic nuclei. And the anterior margin of the thalamus, of course, is the column or the pillar of the fornix. Now, underneath again the hypothalamic sulcus, we have all the hypothalamus, which is part of the wall of the third ventricle. So you see that sort of 1/3 of the wall of the third ventricle, lateral wall of the third ventricle, is hypothalamus, and the rest is pretty much thalamus. The floor of the third ventricle is both hypothalamus and midbrain. Again, anatomy is definition, is nomenclature. You can see here the mammillary body, you can see here the tubercinarium, and then you can see the chiasm anteriorly. Okay, the mammillary body, by definition, divides anteriorly what is hypothalamus and posteriorly what is midbrain. So you see that the floor of the third ventricle, almost half is midbrain. We don't realize this very frequently because we are accustomed to think about this being the floor, because that's what we deal mostly in surgery, since this is pretty much already midbrain itself until, of course, the opening of the aqueduct. We'll have later on a lecture about the third ventricle, and we will return to these important structures. So again, if we come back to this initial image, we know that the thalamus is the center of the brain. The thalamus is the top of the head of the side of the brain stem. And we already know that the hippocampus, together with the fimbria and then the crura of the fornix, go around the thalamus. And we see here the choroid fissure we're talking about. And the choroid fissure, of course, has the choroid plexus along the choroid fissure. So since the foramen Monroe is just anterior to the thalamus, as we already mentioned, there is no choroid plexus in the anterior horn. There's just choroid plexus in the body of the ventricle, in the atrium, and in the inferior horn. The second structure we're going to be putting around the thalamus is the caudate. The caudate also has a head, has a body, and has a tail. And as we emphasized initially, all these structures have this C-shaped format. Now, while the crura of the fornix itself goes medially here, of course, the caudate takes this more lateral position. And in between the fornix, the caudate and the thalamus, of course, we have here the very important vein, the thalamocaudate vein. Here we have the choroid fissure without the choroid plexus, here with the choroid plexus, as you can see. And you see here the foramen Monroe, which is very anterior to the thalamus. And again, posterior wall of foramen Monroe, you have the thalamus with the anterior tubercles of the thalamus, and anteriorly you have the pillar of the fornix. Posteriorly, we have the pulvinar. Okay, medially we have the crura just going around the pulvinar here medially, and laterally, as already mentioned, we have the tail of the caudate goes laterally here, and we can in between, of course, see here the pulvinar. This preparation shows that the caudate has its head, the body, and the tail, and you can see that the tail of the caudate, which we're talking about now, runs along this roof of the inferior horn, goes along the roof of the inferior horn, and get what? It goes and merges with the amygdala anteriorly, while the hippocampus runs along the floor of the inferior horn, as we already mentioned here. Okay, now, in this preparation, we did remove the corpus callosum. We can see again the head of the caudate with the caudate veins here, and we can see the whole fornix dissected. You can see here the crura of the fornix, the body of the fornix, the pillar of the fornix, which is the anterior wall of foramen Monroe, as we emphasized, going towards the mammillary body. From the malleable body, you can dissect, as Professor Ture will be showing later, the mammillothalamic tract, which will go from the mammillary body towards the anterior thalamic tubercle. You know that the fibers from the anterior thalamic tubercle will project in the single. Everything that comes to the thalamus will project to the cortex, and then we'll have along the cingulum here, and of course you have here the circuit of Papez. We're going to be talking about this later, but you can see here that the column fornix runs exactly posterior to the anterior conch. But at this point, I want to show, since this case doesn't have a very big thalamic vein we're seeing here, we can see another bundle of fibers here. This bundle of fibers that runs underneath the thalamocaudate vein is the stria terminalis. What is the stria terminalis? The stria terminalis was described by a neuroanatomist called Johnston, and it's a dorsal extension of the amygdala that is inside here, the anterior part of the uncus. The amygdala has a dorsal extension that it's called the stria terminalis that will end at the so-called bed nucleus of the stria terminalis. And more recently, Heimer and Olmos described that the amygdala also has a ventral extension that will come in through the temporal stem to the substantia nominata or the striatum ventral region. We'll talk about this in the next lecture. And will also end at the bed nucleus of the stria terminalis. That's why these authors came with this concept of extended amygdala, because the amygdala would have a dorsal extension and a ventral extension. And these are not only fibers, because they have neurons in between here. That's why they are called extensions of the amygdala and not simple fibers, tract of fibers. So here the stria terminalis to be seen in this preparation. Now, in this dissection, we did cut the cerebral hemisphere here in order to show that the corpus callosum is indeed the roof of the ventricular cavity we're talking about. Now, we are very accustomed to think about the corpus callosum along its midline, and we can see that here we have the rostrum of the corpus callosum. In anatomy, everything that is facing down we call rostrum. Then you have here the knee or the genu of the corpus callosum, and then you have the body that is continuous with the splenium of the corpus callosum. But the corpus callosum has all these fibers. Of course, it's our main commissure, and you can read that the corpus callosum has around 200 million fibers, and these fibers open in every direction. So the best way that I found to describe the three-dimensional anatomy of this corpus callosum, it's to think about a butterfly, and this would be the body of the butterfly, while the fibers that are opening up in all directions would be the wings of the butterfly. But we have to imagine the butterfly with the wings going up and down to understand the complex anatomy of these fibers that are opening in all directions. So here you can see again now the caudate, the choroid plexus along the choroid fissure, and you have to know that everything everything medial to the choroid plexus is fornix, is the crude of the fornix that is wrapping the thalamus here. So if you cut here, you'll be cutting initially the crude of the fornix, and then you'll reach the thalamus. The surface that you can see in the lateral ventricle, in the body of the lateral ventricle, the thalamic surface is in between the choroid fissure with the choroid plexus and the caudate. This small part here, neuroanatomists call this surface the lamina affixa, because it's exactly here where the diencephalic structures are attached to the telencephalic structures. This is the only surface from the telencephalon that we can see here in the lateral ventricle that is a diencephalic surface. This is called the lamina affixa, this part of the thalamic surface here. So we talked about the fornix and we talked about the corpus callosum. Now for us, I didn't talk about the mass intermedia that some people do have, others don't have, that puts together both thalami in the mid-thalamus here. Now, very important for us neurosurgeons to understand the relationship between the fornix and the corpus callosum. you see that the crura of the fornix is very much attached to the inferior surface of the corpus callosum here. And then you have the body of the fornix here, and then you have the column of the fornix getting inside the hypothalamus, as we already saw, being the anterior margin of the foramen Monroe. Now, if we see from the bottom, we can see that both of these crura of the fornix, both crura of the fornix are attached to this inferior surface of the splenium, and in between, you have fibers that put them together, so they are called forniceal commissure or hippocampal fornissure, because functionally they put together both hippocampi. Of course, here you have the pulvinar, and of course, here you have the choroidal fissure, the posterior part of the choroidal fissure open here, as you can see, because we are pushing the brain upwards. Now, attached both to the corpus callosum and to the fornix, you have the septum pellucidum. The septum pellucidum is constituted by two leaflets, two leaves of two membranes, with each one attaching to each fornix. So the septum pellucidum is attached to the body of the fornix, to the genu of the fornix, and to the rostrum of, to the body and to the genu of the callosum and to the rostrum of callosum, here anteriorly and basally, and posteriorly and basally is attached to the fornix. And of course, the rostrum callosum then ends exactly where the crura of the fornix is attached to the inferior surface of the splenium. So if we come to this point where they get together is exactly this point. And again, anatomy are definitions. Anatomy are definitions. From here, anteriorly, you have the body of the fornix. From this point posteriorly, you have already the atrium. You have already the atrium. To understand more this anatomy, as an exercise, let's understand the cavum pellucidum and the cavum vergae. I emphasize that the septum pellucidum is constituted by two leaflets, okay, with each one attaching to each fornix. So if the fornix are separated embryologically, these leaflets will be also separated. And there will be a cavity in between these two leaflets, which is called the cavum. Now, we define the cavum of septum pellucidum, the part of the cavum that is anterior to the anterior horn. And we define the cavum vergae, the part of the cavum that it's posterior, to the foramen Monroe. Anterior foramen Monroe, we have the cavum pellucidum, and posterior to the foramen Monroe, we have the cavum vergae. So you understand that you have this compartment here that will lead inside the third ventricle because both fornix are separated originally. And that's why both leaflets of the septum pellucidum are also separated. So here again, you have the fornix, and here you have the septum pellucidum. You see that anteriorly, it will be attached to the most anterior part of the fornix until to the rostrum of the callosum, until be anteriorly to the foramen Monroe. At the level of the foramen Monroe, you can see here attached to the pillar of the fornix that we go towards the mammillary body, and posteriorly you have attached already along the body of the ventricle, and you can see here the crura of the fornix already going more lateral. If we understand this anatomy of the cavum, we can understand the relationships of the septum pellucidum with the fornix, of course. So if we come back to this dissection here, we have to know already by what we talked that the anterior horn is anterior to the thalamus and has as its anterior wall the genu of the callosum, as its floor the rostrum of the callosum, as lateral wall the head of the caudate, and its medial wall the septum pellucidum. So the septum pellucidum is both the medial wall of the anterior horn and of the body of the ventricle. Posteriorly to here we have the body of the ventricle, and posterior to here we have already the atrium, okay? And you have, of course, the body of the caudate running along the lateral wall of the body of the ventricle, and then goes more laterally here at the level of the atrium. The anterior wall of the atrium would have the crude of the fornix medially, of course, the choroid flexus here, and more laterally, the pulvinar, this part of the surface of the thalamus, which altogether is called the lamina affixa, because it's where the diencephalic structures are attached to the telencephalic structures. More posteriorly, we have here the emergence of the insertion of the tail of the hippocampus in the medial wall of the atrium. You have the bulb of the callosum. You know that you have the forceps major inside the splenium and the forceps minor inside the genia of the callosum. And posterior to it, you have the calcar avis, which is the inner aspect of the calcarine fissure. This is why this is gray matter. Here we can see now the base, the floor of the atrium, and you can see that has this very triangular shape. It's very flat, and this is called the collateral trigone because the collateral sulcus is running exactly underneath here. Again, you have the hippocampus here with its tail at the most anterior part of the medial wall of the atrium. You have the bulb of callosum just posteriorly. You always have this upper angle of this trigone area here, which again is the collateral trigone. If we go more anteriorly here, we can see that this flat surface becomes round. It's a prominence and becomes the collateral eminence. So while the collateral trigone is very flat, the collateral eminence is already round and runs just parallel, just next, just lateral to the hippocampus as shown here. And with both of them, of course, forming the floor of the inferior horn. So we already talked about all these structures around the lateral ventricle here. Let's talk about everything that is in between now and the roof of this ventricular cavity. If we see from above, we can see the insula here laterally, and we can see that in between the inferior horn inferiorly and the anterior horn and the body of the ventricle and the atrium, you have a very well delineated block here that Dr. Rotten called the central core of the brain. You can see then we'll have a special lecture of this given by Eduardo, who studied that at Rotten's lab. But you can already have in mind here that the anterior half of the insula is related with the body, with the head of the caudate, and the posterior half is related both with the body of the caudate and with the thalamus, with the foramen Monroe a little bit anteriorly to the midpoint of the insula. So you can see that this is a very well-defined block that it's called the central core of the brain, and which harbors inside the internal capsule. You see here the putamen. The internal capsule is the internal capsule of the putamen, and the external capsule is the external capsule of the putamen. Dr. Turia will be talking about this more shortly, but as you know, you have an anterior limb, you have a genu that is just next to foramen Monroe, you have a posterior limb, you have a retrolentiform part and a sublentiform part. With everything, this will open these fibers and will cover the ventricle. So when we expose the insula now, you have to have this X-ray vision. You have to know that the anterior part of the insula harbors it's related with the head of the caudate. The posterior half of the insula is related with the thalamus, and up here with the body of the caudate. So the third ventricle is just related with the posterior part of the insula. I used to say that the anterior horn and the third ventricle are the same level of this building, just as the atrium that is posterior to the central core here. So the atrium, the third ventricle, and the anterior horn are the same level of this building, while the body of the ventricle is one level above, and inferior one level superiorly, one level above, and the inferior horn is one level below this bigger floor of this big building here. Now, the internal capsule has this fan shape, since the fibers are coming from the corona radiata and goes towards the cerebral peduncle, and these are the most lateral fibers that are covering internally, of course, the putamen. And as you can see here, which if you have the putamen left, again, anatomy's definitions, everything that is superiorly to the putamen is called the corona radiata. And if you remove the putamen and you expose the global pallidus, of course, the fibers from the corona radiata that are now internal to the putamen are the so-called internal capsule itself. Again, the fan-shaped format of the internal capsule, which is more lateral, and the inner fibers of the internal capsule are the thalamic radiation, are the fibers that are arising from the thalamus and will project, of course, in the cortex. This particular dissection was done by Leonard Heimer in Charlottesville, Virginia. At the time, I had the privilege to study a little bit with him. Again, let's remember that the internal capsule then have this fan shape, and it divides the striatum, only the dorsal striatum. So medially, we have the caudate, and laterally, we have the putamen with the ventral striatum left. It's what we can see in this beautiful dissection done by Richard. You can see that the dorsal striatum was divided in caudate and in putamen, and the ventral striatum is right here intact, was not divided because the fan shape of the internal capsule. And ventral striatum is pretty much synonym of accumbens. The accumbens nucleus is really harbored here inside the ventral striatum. Let's finish with some lateral dissection just to introduce what Dr. Ture will be talking about. I'm sure that anatomy is very, very important to have repetitions in order to fix the most important concepts. If we remove initially the surface of the brain, we can see the superior longitudinal fascicle, and if we dissect this more carefully, we can see that we have an horizontal part, a vertical part, and inside this fissure, the more inner fibers would be a complete C format fibers that would constitute the so-called arcuate fascicle, which is considered a different fascicle from the superior longitudinal fascicle. In chartography, we can see this much better, these fibers that are more difficult to dissect in the regular brain. If we remove the surface of the insula, we can see the extreme capsule, which is nothing more than the U fibers of the insular surface. If we remove a little bit more, we can see the external capsule, a little bit of claustrum here left, and we can see that external capsule is continuous with these two very important bundle of fibers, the most anterior one being the uncinate fascicle, and the most posterior one being the occipital-frontal fascicle that were left here. At this point, we did remove the posterior, the dorsal part, posterior part of the external capsule, exposing, of course, the putamen with the corticostriatal fibers coming here inside the putamen. The corticostriatal fibers, conceptually, they are not part of the internal capsule because they don't go medially, they don't go towards the peduncle. They just end inside the putamen. You have corticostriatal fibers coming both to the putamen and medially to the caudate as well. And here it was left the occipital-frontal fascicle that Ture was talking more about this, and anterior you can see here the so-called uncinate fascicle. If we remove the putamen, we can see the globus pallidus already with the internal capsule we just showed. And if we look here, we have a pretty well-defined region, a compartment that has as its posterior wall the globus pallidus itself and the anterior commissure, at its superior aspect, as its roof, the fibers of the anterior limb of the capsule, as its base, its floor, the anterior perforate substance itself, and medially you have the ventral striatums, you have the accumbens, and anterior, of course, and superior, you have both the occipital-frontal fascicle and the uncinate fascicle. Of course, through here, you have entering the lenticular-striate arteries, the perforators, that comes inside this region. This region, it was used to be called by substantia nominata. I know this is the name that Dr. Ture uses. And Heimer used to call this the ventral striatum or ventral pallidostriatum region. Recently, I heard Michael Lawton referring to this region as the anterior perforate substance region, which I think that it's also a good name. Medially, we're going to be seeing later that this region is continued with the septal region, and laterally with the so-called temporal stem. Again, here you have here the perforators. You have this region we're talking about. And now, if we remove the occipitofrontal fascicle and the uncinate fascicle, we're seeing better the anterior commissure here, running here at the base of the globus pallidus. And the anterior commissure fibers, they open here, constituting the most superficial fibers of the so-called sagittal stratum that covers the ventricular cavity. We can detach the anterior commissure from the so-called channel of Gratiolet here, and if we cut and peel these more superficial fibers, the fibers we will have underneath will be mostly pertinent to the optic radiation. As Ture emphasized, you cannot really separate all these fibers. These fibers go anteriorly, becoming the so-called Meyer's loop, and then go posteriorly. And of course, here you have again the anterior commissure. And if you put this anterior again, you have the superior extension of the amygdala blending with the globus pallidus. And here the beautiful channel of Gratiolet with the accumbens nucleus medially here. Now, this preparation is very interesting because in this side you have intact, in this side you have dissected, you have here the accumbens, you have here the anterior commissure, just going posterior to this structure I want to talk about now, which is the ansa peduncularis. You have here the amygdala, and the ansa peduncularis is constituted by all amygdalofugal fibers together. Fibers that go to the septal region, fibers that go to the thalamus, and fibers that go to the hypothalamus. It turns out that the most superficial fibers that go to the septal regions, when seen here in the intact anterior perforate substance, they look like a crest, a small ridge here, that it's called the diagonal band of Broca. So this is the most superficial part of the ansa peduncularis that it's going towards the septal region that you can see better. We're going to be talking more about this in the next lecture, which is constituted by the paraterminal gyrus that harbors the septal nucleus, and anterior, the posterior parafactor gyrus, and anterior parafactor gyrus, just posterior to the so-called cingulate pole as named by Professor Yassagil. And of course, all this anterior to the anterior commissure. Another beautiful dissection done by Richard here, you can see that the anterior commissure is passing by both parts of the fornix. We usually think about the fornix at this post-commissural part of the fornix that it's going towards the mammillary body. But the fornix also has this anterior portion, which is the precommissural portion of the anterior commissure that goes towards the septal nuclei. So you see here the anterior perforate substance, the anterior commissure just passing in between. And in this side, we can see this branch of the anterior commissure that Theodor Meiner called the olfactory branch. Theodor Meiner divided the anterior commissure in an hemispheric part, in an olfactory part, that it's beautifully dissected here by Richard and coming to the olfactory tubercle. In this preparation, you can see all the layers we talked about, the superior longitudinal fascicle. You can see here the sagittal stratum. You can see here, of course, the insular surface, the extreme capsule together with a little bit of capsule, and the external capsule, and here the uncinate and the occipitofrontal fascicle. And in this window, you can see that underneath the so-called sagittal stratum, you have another bunch of fibers that have a different orientation. They are, and these fibers are coming from this posterior part of the splenium. These are the tapetum fibers. If we go, for example, for the Gray's Anatomy book and we look for tapetum, we see that the tapetum, at least there, is defined as the fibers that run underneath the optic radiation. It makes sense because tapetum means carpet. So this is the carpet that runs along the inferior aspect of the optic radiation, just at the roof of the ventricular cavity. Let's end this removing now the fusiform gyrus, that emphasizing that the fusiform gyrus is the floor of the inferior horn and of the atrium. The anterior part of the fusiform gyrus is the floor of the inferior horn, and the posterior part of the floor of the fusiform gyrus is the floor of the atrium. We're going to be talking about this showing some surgery in the last lecture we're going to be giving. But inside the fusiform gyrus, we have the inferior longitudinal fascicle. And if we proceed our dissection, we'll be coming to the roof of the inferior horn. You can see here the optic radiation with the Meyer loop coming here. And you can see here, under most posterior, you would have the tapetum underneath the optic radiation fibers. And you can see that the optic radiation fibers, they come not only from the lateral geniculate body, but they come from all this posterior part of the thalamus. And that's why old anatomists used to call this optic thalamus. This posterior part of the thalamus is called by some old anatomists the optic thalamus, because from here they arise all this optic radiation fibers. If we remove this, we can see the upper part of the roof, and we can see here beautifully the tail of the caudate, that it's coming in the roof of the inferior horn and go towards the amygdala. Remember that the amygdala is anterior to the head of hippocampus that was removed, but also goes superior and covers a little bit the head of hippocampus. And of course, you have here this posterior extension of the amygdala, which is the stria terminalis, described by Johnson, as already mentioned, and medially the optic tract going towards the region of the lateral geniculate body. Here you can see again and understand that the tail of the caudate goes along the roof of the inferior horn and will blend with the amygdala. Will blend with the amygdala, just as the globus pallidus will also blend at the amygdala, as we showed. So we can understand this region here. Again, the amygdala going superior. So the amygdala will be receiving the head, the tail of the caudate here along the roof, and will itself go up and blend with the globus pallidus, with all these structures blending around this region here. Okay, and let's remember that the amygdala then will be continuous then with the globus pallidus at this region, and just constituting anterior to the head of hippocampus sort of a peduncle that will insert this part of the brain in the most lateral aspect of the basal forebrain. And just posteriorly to this peduncle, you have the choroid fissure. So you have the thalamus, which is the head of the brainstem, just attaching to along the line over the subiculum, okay, and just where the ambient system is. You can understand here in these two dissections very well this complex anatomy, And just anterior to this, you have the peduncle that comes from the amygdala and will be then continuous with the most lateral basal forebrain. And that we understand this as this controversial concept of temporal stem that we'll be talking in the next lecture. I'll finish here for the sake of time. And I want to say again, thank you very much to Rod, to all Seattle for Seattle Science Foundation for this new opportunity.